Hello everyone. Today we'll discuss the UPHESC Biochemistry 2021 previous year question paper, and this is part 2, which is subjective and consists of 70 questions. The part 1, which is GK part, has already been discussed and is available on the on the channel. Please go look for it. Now, question number 1: name an inhibitor of endocytosis. So options are chlorpromazine, option E64, option C PMSF, or option D benzamide. So the right answer is chlorpromazine. Next question is the fatty acid with how many carbon atoms can enter mitochondria without the help of membrane transporter. So fatty acid during oxidation has to pass from cross this membrane, mitochondrial membrane, like from cytosol, it has to move inside the mitochondria. And during this, if the carbon chain length is equivalent to 12 or less than 10, then it can pass directly. Or if it is longer than that, if it is 14 carbon or more than 14, if it is 14 carbon chain length or more than 14 carbon, then it needs a transporter. So that it needs carnitine for its transport. But fatty acid with length of carbon 12 with length of 12 carbon or less, they can cross this mitochondrial membrane directly. So answer will be B, 12 or less. Next question is coattamer coated vesicles mediated vesicular transport from ER to Golgi is. So it is selectively towards Golgi apparatus, it is selectively towards ER, or it is non-selectively, or it is semi-selectively. So the answer is it is non-selective in nature, and when during transport of protein, so ER from the ER, the vesicles are burned off and then they go to Golgi bodies. So here these are coattamer coated, so they are COP2 coated. The vesicles which are meant for ER to Golgi body, they are COP2 coated, and here the interaction is receptor mediated, so it is a non-selective transport from ER to the Golgi. Now facilitated diffusion takes place through specific channels, specific pore openings, specific carrier proteins, or non-specific carrier proteins. So the right answer is, in this case, specific channel. Facilitated diffusion takes place through specific channel as well as specific carrier proteins. So both options are correct. Now, which substances are transported through facilitated diffusion? Galactose, urea, alcohol, or oxygen. So the right answer is galactose. Now, sodium and potassium pump work as so symporter, uniporter, antiporter, or A and C both. So the right answer is antiporter. So in antiporter means one is transported in one direction and the other is direction in the opposite direction. Symporter means traveling along in the crossing the membrane in the same direction. Symporter is like transport of glucose along with the sodium ion is an example of symporter. And sodium potassium pump work as antiporter. Now the next question is the N-linked glycosylation occurs in which amino acid sequence? So N-linked and O-linked glycosylation, they are post-translation modification, and N-linked glycosylation, it takes place in ER, and there is a particular sequence present in the protein where this N-linked glycosylation takes place. So N-linked glycosylation happens in asparagine, on asparagine. So on the amino group of asparagine, there is a glycosyl group attached. There is a sugar moieties attached. So this asparagine has to be present in these amino acid sequence in order to be sequenced. Only then this asparagine will be glycosylated if it is present in this sequence. Asparagine, X, serine or threonine, X could be any amino acid. Now the next question is the lysosomal sorting signal is N-acetyl glucosamine, mannose-6-phosphate, RAN-GTP, or ribose-6-phosphate. So the right answer is mannose-6-phosphate. So while protein synthesis and the sorting to various organelles, from Golgi, when it is translocated from Golgi body to different organelles. So mannose-6-phosphate, this acts as a sorting signal for lysosomal enzymes, which means that those protein which will have this mannose-6-phosphate on their uh, protein, they will be translocated to lysosomes. Largest intracellular cell organelle in a cell is, so in a cell, nucleus is the largest cell organelle. Now, which of the following glycosidic linkage is present in cellulose? Alpha 1,4, beta 1,4, alpha 1,6 or beta 1,6. So in cellulose, beta 1,4 glucose, this is the linkage. Which of the following is not present in cholesterol? Hydrocarbon tail, OH group, or steroid, or sugar? So sugar, there is no sugar molecule. In steroid, you will see there are chains like this, that is called as the steroid nucleus. There are 4 chains, A, B, C and D. 4, 3, 6 carbon, and then there is one 5 carbon, 5 carbon ring, and to these there are different OH group attached and there are fatty acid attached, hydrocarbon tail. So there is no sugar in the cholesterol. Cellular protein folding is not assisted by molecular chaperones. Chaperones do have a role in protein folding. They keep the proteins in a native state until they are folded properly. Protein disulfide isomerase, it induces disulfide bond formation in the protein. Peptidyl propyl isomerase and the proteasome. So in this case, proteasome what it does, it actually degrades the protein. It has a role in degradation of the protein. So answer D will be the right answer. Now, which of the following disease is caused by human prion protein? Kuru, Alzheimer's disease, Parkinson's disease, or chronic wasting disease? So, Kuru is a disease which is which is caused due to human prion protein in human. Now, which of the following is not an eicosanoids? Leukotriene, prostaglandin, plasmalogen, or thromboxane? So, right answer is plasmalogen. Next question is, a group of protein, the misfolding which causes it to aggregate and produce the neurodegenerative symptoms of transmissible spongiform. It is spongiform actually, it's not, it is spongiform. Encephalopathies and related disease is known as. So prion proteins are responsible. So prion protein, they are present in membrane. They attain a different configuration. There's a change in their secondary structure of these prion protein, and they when they are disordered, they form this beta amyloid or beta sheet structure which produce aggregates. So then they cause this disease. So prion proteins are responsible for these neurodegenerative symptoms of transmissible spongiform encephalopathies. And other prion disease in different, for example, as we have talked, Kuru in human, and in other animals also it causes disease. The peptide bond C and bond is, so C and bond, if we see, the amide bond is like this. This is the amide bond, but this amide bond, these electrons of this double bond resonate over here, so the bond is like this, and that's why this bond is smaller than the normal single CN bond. So it is shorter than the C alpha N bond. Now the Lineweaver-Burk plot of enzymes reaction slope is denoted by, so it is denoted by Km by Vmax. So in Lineweaver-Buck plot, it is plotted between 1 by S and 1 by V. So graph is like this. So here it represents minus 1 by Km. So the x-axis, its intercept on x-axis, it represents minus 1 by Km, and it represents 1 by Vmax. And the slope is 1 by Vmax by 1 by S. So it will be 1 by Km. So the slope will be Km by Vmax. This will be the slope. Now, which of the following inorganic ion serves as a cofactor for enzyme pyruvate kinase? So pyruvate kinase potassium ion act as a cofactor. Which of the following absorb light at 340 nanometers? So NADH act as NADH and not NAD plus. NAD plus does not absorb any light at 340. So NADH absorb light at 340 nanometer. Mutarotation of glucose is an example of. It is an example of acid-base catalysis. The other options are covalent catalysis, catalysis by proximity and orientation effects, catalysis by preferential binding of the transition state complex. So this is the structure of glucose, and in solution it is mostly present. Let's say glucose, yes, glucose, it is present in solution in a pyranose form. So this, it could be present in an alpha or beta form. If the OH is above, it will be beta form. So in solution, it will be first, it could be converted to linear form, and then it could be from this beta form, it can be converted to a alpha form. So this is called as mutarotation, and in this, because it needs an water molecule, this is called as acid-base catalysis. Now next question is enzyme catalyzed group transfer reaction mechanisms, in which all substrate must combine with the enzyme before the reaction could occur and products be released, are known as. So this is known as sequential reaction, where all substrate have to bind first, combine with the enzyme in order for the reaction to start, and then the products are released. Now the next question is enzyme which can join two chemical groups are called. So any molecule which can join two molecule is called as ligase. Ligase means which breaks a double bond, and hydrolase, any reaction hydrolase is it also causes lysis of a bond, and it involves a water molecule. Oxidoreductase, where the oxidation and reduction of a oxidation and reduction takes place during the reaction. Zymogens are, options are enzyme acting as antigen, no, they are enzymes, active enzyme precursors, inactive enzyme precursor, or enzyme inhibiting by ligand binding. So zymogens, when enzymes uh, are present initially before their initially they are present as inactive, in the inactive form. For example, during digestion, the many of the enzymes in the intestine, they are present in an inactive stage. When there's no work, when they don't have any food to digest, so they're present in inactive form, and they're called as zymogen. So when there's a food in the intestine, they will be activated by trypsin, chymotrypsin, or by the acid present in the stomach. So there are various ways of activation of the enzyme, and then these inactive enzymes are converted to the active form. So inactive form of the enzymes are called as zymogens. Which of the following is most appropriate for ribozymes, which are RNA molecules that can catalyze biochemical reactions? So ribozymes, it means they are RNA molecules which have catalytic activity. So we have to find which among these following statement is the most suitable for them. They have KM, they have both KM and Vmax. They have Vmax but do not have KM. They do not follow Michaelis-Menten kinetic. So among this is the most appropriate statement. They have KM as well as Vmax. Next question is Koshland proposed dash which model? So lock and key model, refractive index, induced fit or Benjamin fit. So the right answer is induced fit model. Koshland produced the induced fit model for the enzyme and the substrate binding. Now, what is an enzyme called which is activated by cleavage? So, as I have already told, it is zymogen when it is present in active form, and then further it is converted to active form. It could be by different means. So, one could be by the cleavage of some part of the protein. Which amino acid can form disulfide bond? So, cysteine is the only molecule. It has the SH group, and these two SH group, when they are oxidized, uh, these two SH group, they form this disulfide bond by the help of this. If we talk in terms of within the cell, it is catalyzed by protein disulfide isomerase. This we have talked earlier. So cysteine is the only amino acid which undergoes disulfide bond formation. So the next question is, which of the following technique is not useful for the molecular weight determination of the native protein? So it is asked not useful. So native page is used for determination of the molecular weight because here the protein are separated on their molecular mass. Ultracentrifugation is also used because here in the column the proteins or macromolecule, they are separated based on their molecular weight. And size exclusion chromatography, it is a chromatography where the analytes are separated based on their molecular weights. So SGS page is not used. So right answer is SGS because it is asked which is not useful. So SGS page could not be used for molecular weight determination because in this the protein uh, is um, is converted to its subunit. It is denatured to its respective subunit. So the molecular weight of the native protein could not be determined. It can only determine the molecular weight of the subunits, and it could not tell the how many subunits are present in the native protein and what will be its molecular weight. So next question is, which of the following is not used for reducing the disulfide bonds in STS page? So, um, for the denaturation in STS page, the disulfide bonds are reduced to SH. So, it is done by BME. BME is most commonly used. DTE also act as a reducing agent, and the among these protein disulfide isomerase. So, this is not used. So the right answer will be protein disulfide isomerase. So as we have talked earlier, PDI, it induces disulfide bonds. So it is not used in the in reducing the disulfide bond in STS page. All these three, BME, DTE, and TSEP, ris-2 carboxyethyl phosphine, are used in the STS page for disulfide bond reduction. Now next question is, a protein exists in mono, di and triphosphorated form. Which method can be used to identify each form? So if a protein has mono, di or triphos, so it means there will be some difference in their molecular weight and some charge. So it could be either be identified through isoelectric focusing. In isoelectric focusing, the protein are separated based on their charge, so they will have different pi, and that's why it could be identified through IF, isoelectric focusing, and as well as the mass spec, but it could not be determined through native page or the size exclusion chromatography. Now so in case A and B both are the right answer. 2D polyacrylate amyl gel electrophoresis, that which is called as the 2D page, separates protein based on pi or the molecular mass or hydrophobicity. So in 2D page, 2D means two dimensions. So first First, the protein are separated based on their pi. So you add protein over here, and based on their pi, they move on the anode and the cathode side. They are separated, and then this strip is placed over the SDS page. Then the protein are separated in this direction based on the in the SDS page based on the molecular mass. So in 2D page, first it is separated based on their pi in isoelectric focusing, and then they are separated based on their molecular mass using page. So both A and B are the right options. Now, in several human diseases, protein form aggregates and deposit as amyloid forms. Now, which are rich in beta sheet structure by using CD spectroscopy? At what wavelength characteristic peak of amyloid will be observed? Options are 220 nanometer, 208 nanometer, 280 nanometer, or 218 nanometer. The right answer is 280 nanometer. So the amyloid structure are observed at 218 nanometer. Next question is, plasmid DNA is resolved by agarose gel electrophoresis, polyacrylamide gel electrophoresis, SDS page, or 2D gel electrophoresis. So I think everyone knows they are separated by agarose gel electrophoresis. Optical rotation as a function of wavelength is known as circular birefringence, circular dichroism, ellipticity, or the optical rotatory dispersion. So the option D is the right option, optical rotatory dispersion. Now, next question is, in mitochondria, conversion of pyruvate to acetyl-CoA is. So, option are oxidative decarboxylation, reductive decarboxylation, oxidative carboxylation, or reductive decarboxylation. So, uh, pyruvate is the end product of glycolysis, and before entering the Krebs cycle, it is converted to acetyl-CoA, and then it enters the mitochondria, and it enters into the Krebs cycle. So pyruvate, it is a 3 carbon molecule, and acetyl-CoA is a 2 carbon molecule. So in this there is a release of CO2. So it is a decarboxylation reaction first of all, and if you remember, in this process one NADH is generated at this step. So this means that it is an oxidation reduction is taking place. So while this pyruvate is converted to acetyl-CoA, it is oxidation is taking place. So it is an oxidative decarboxylation. The right answer is A. Now the next question is the current sequence of fatty acid degradation. So when fatty acid degradation takes place, this 12 carbon chain is converted to in every cycle it is shortened by 2 carbon molecule. In every it will be there will be steps of reactions, and then in every cycle there will be release of 2 carbon acetyl-CoA, and it will be converted to 10 carbon molecule. In each cycle it will be shortened by 10 carbon. For example, if we have a palmitate chain 16 carbon, so it will take total 7 cycles for the oxidation of this 16 carbon palmitate molecule by the beta oxidation pathway. So in this this sequence of reaction in release of one in this cycle, there are 4 steps. The 4 steps is first oxidation, then hydration, then oxidation at the beta carbon, and then the final is cleavage, where this 2 carbon molecule is released. So the right answer is A, oxidation, hydration, and oxidation and the cleavage. Now the next question is, which of the following amino acid is non-essential amino acid? So histidine, lysine, methionine, all these are essential amino acid. Glycine is glycine, alanine, valine. These are non-essential amino acid. Now the next question is, dash is involved in protein degradation. So cytochrome C, molecular chaperone, peptidyl propyl isomerase, or ubiquitin. The right answer is ubiquitin. So if you know that ubiquitin, there is a method of degradation of proteasome. So it is ubiquitin mediated degradations, where the ubiquitin are first ligated to protein, and then which dictate ubiquitin is ligated to protein which degrade it, which mark it for degradation by the proteasome. So answer is B, ubiquitin. The next question is the enzyme involved in the last step of urea cycle is. So urea cycle where in the ammonia is converted to urea in the liver, and this is known as the urea cycle. So in this the last step has been asked. So the first carbamyl phosphate synthetase 1, this is first step actually. Ornithine transcarbamylase, arginosuccinase or the arginase. So arginase, this is the last step where in this which urea is released. So the right answer is arginase. This is the last step of the urea cycle. The arginase catalyzes the last step of the urea cycle. Now avidin has a very high affinity for biotin. Which of the following reaction would be blocked by addition of avidin? So actually biotin act as a cofactor for many reactions. It acts as cofactor for reaction where there is an addition of CO2 molecule or one carbon molecule. So glucose to pyruvate, there is no carbon dioxide addition. If you see malate to oxaloacetate, it is also a 4 carbon, it is also a 4 carbon molecule. And here if you see pyruvate to glucose, if we see this, this is a part of gluconeogenesis. Gluconeogenesis. So in this, first the pyruvate is converted to oxaloacetate and for transport from mitochondria to cytosol, and then it is enter the in the cytosol. So this here this there is the addition of carbon dioxide. So this reaction will be affected by the if which by the addition of iodine, because iodine will interact with biotin and it will capture the biotin. So there will be deficiency of biotin. So in that case, carboxylation reaction will be affected, which takes place during this pyruvate to glucose conversion. Now classification of living organism into three groups: monotelic, deuteric, or uricotelic have been done on the basis of. So the options are the way they synthesize their amino acid, the mechanism of deamination reaction, whether the amino acid are predominantly glucogenic or ketogenic, and the right option is A, which is the way they excrete the excess nitrogen resulting from the metabolic breakdown of amino acid. I think everyone knows this, that ammonia, because uh, is toxic. So living aquatic animals, there's no shortage of water, so they liberate ammonia, and the birds and reptiles, they are uricotelic in nature, and rest terrestrial mammals are ureotelic. So this classification is based on how they release ammonia. So mammals, including all the terrestrial mammals, as we were talking earlier, ammonia is first converted to urea, and then this is released. So those organisms are called ureotelic. So this classification is based on how the nitrogen waste is eliminated from the body. Now in next question, in vivo fatty acid are mainly catabolized by alpha oxidation, gamma oxidation, delta oxidation. So the main method, the main mechanism by which fatty acids are catabolized inside mitochondria is beta oxidation. Alpha and gamma, the omega oxidation, these are minor pathways. Both PEP carboxylase and Rubisco are found in the mesophyll cells of C3 plants, C4 plants, C4 plants, or the CAM plants. So the right answer is CAM plants. Next question is complete conversion of glucose in the presence of oxygen into carbon dioxide and water. The release of energy is called as. So this is called as aerobic respiration. Complete conversion of complete means complete oxidation of glucose and in presence of oxygen. So in presence of oxygen, it is called as aerobic respiration. In living cells, de novo pyrimidine biosynthesis is regulated by feedback inhibition or by the concentration of purine nucleotide or by the feedback inhibition as well as the concentration of purine nucleotide. And then then the D option is a mechanism different from these. So the right option is C. In living cell, pyrimidine de novo biosynthesis. Pyrimidine biosynthesis by the de novo pathway is regulated by feedback inhibition as well as by the concentration of the purine nucleotides. Now, the next question is the hormone gastrin is secreted by. So, when there is a food in the stomach, so there are different types of cells, G cell, oxyntic cells present in the stomach. They secrete different hormones. So, they secrete gastrin in response to food, and this is secreted by the G cells of the stomach. WBCs, which release heparin and histamine. So there are different type of white blood cells. Neutrophils, eosinophils, and monocytes. So basophils and mast cells are responsible for release of heparin and the histamine. So among the basophil is the type of WBCs which release these inflammators. A percentage of cholesterol in cell membrane is, so the options are 43.3%, 23.3%, 77.3%, or the 33.33%. So it is 33.33%. During peristalsis, depolarization of the membrane is caused by sympathetic nerves, parasympathetic nerves, norepinephrine, or the epinephrine. The right answer is parasympathetic nerves. Now, bile salts helps in the absorption of monoglyceride, proteins, glucose, and minerals. I think everyone knows that bile cells helps in the digestion of fats and lipids, and they're secreted by liver, and the secretion is poured into the pancreas, and where they help in the first digestion of lipids and then their absorption in the small intestine. So monoglyceride, which are actually fats, so it helps in their absorption and digestion. The normal value of hemoglobin in newborn babies. The options are 20 gram per deciliter, 25 gram per deciliter, 17 gram per deciliter, or the 14 gram per deciliter. So the right answer is 20 gram per deciliter. Next question is, which substances activate the platelets? So options are ADP and thromboxane A2, NADP and thromboxane A2, option C ADP and NADP, and option D FAD. The right answer is ADP and thromboxane. Now the next question is, scab is also known as, so internal blood clot, external blood clot, or internal bleeding or external bleeding. So scab is actually the external blood clot which forms when there is a cut. So then the clot form is known as the scab. In the digestion lipid, the phospholipase A and B is activated by option A secretin, option B lipase, option C trypsin, option D pepsin. So in the right answer is trypsin. As we were discussing, they are present, the enzymes are usually present in their inactive form, and when they are required they are converted to their active form. So there will be some stimulus. So for the activation of phospholipase A and B, trypsin leaves a part of their inactive part of it and convert them to active phospholipase and B in the intestine, while when digestion of lipid has to be done in the intestine. All of the following is a protein misfolding disorder except tuberculosis, cystic fibrosis, Alzheimer's disease, or the Creutzfeldt-Jakob disease. So tuberculosis is the right answer. It is caused by it is a bacterial disease caused by the Mycobacterium tuberculosis. Other disease, they are related to protein misfolding. Now the next question is, serum acid phosphatase is a marker of prostate cancer, melanoma, rickets, or the gallbladder cancer. The serum acid phosphatase is a marker of prostate cancer. The next question is, the functional enzyme present in plasma is option A lipase, option B amylase, then option C plasminogen, or option D alkaline phosphatase. So the functional enzyme present in plasma is plasminogen. Next question is, all water soluble vitamins are excreted through urine and are not stored in the body. Niacin, vitamin C, vitamin B6, or vitamin B12. So all these are options, water soluble vitamin B and C. Vitamin B and C, both B complex means they are water soluble and not stored in the body, but there is an exception, which is vitamin B12. Now, one of the following is not a point of care test. So plasma protein plasma glucose, plasma electrolyte, or the arterial blood gas analysis. Point of care test is, it means that those tests which can be carried out on the bedside of a patient. So all these plasma, blood glucose level, blood gas analysis, or the plasma electrolyte level, these can be determined. They are the point of care test. But the plasma protein, they need a certain amount of experiment and time, so it is not a point of care test. Now, nyctalopia, it can occur due to the deficiency of nyctalopia. Nyctalopia is actually night blindness. So we all know it is caused by the deficiency of vitamin A. So night blindness when the person could not have a deficiency in the vision during night. So one cannot see in the dark. Now the here in this question, it is asked to do match of the following. So here we have it is given some receptor or the molecule name, and here are given the molecules or the enzymes related to it. So ApoB100, it is actually a receptor for LDL. These are all the example of lipoprotein complex. APB100, it is an receptor for LDL. So A match to D. HDL is high density lipoprotein. So enzyme LCAT, HMG-CoA. It is HMG-CoA has role in cholesterol synthesis, and which amyloid is an intermediate of cholesterol. So HMG-CoA is involved in the amyloid synthesis. Okay, what does HDL does? HDL is high density lipoprotein. It carries HDL is considered as a good cholesterol. It carries lipid or the cholesterol from the heart or the peripheral tissues to the liver. That's why it is considered as a good cholesterol. So what does this LCAT does? It transesterifies lipids from the peripheral tissue, for example heart, to the HDL molecule. So that's why this HDL is linked to enzyme LCAT, and the rest cholesterol ester is linked to ACAT. Now, according to this, option C will be the right answer. Now, given below are the two statements. One is labeled as assertion and the other is reason. So, assertion is type 2 diabetes is a metabolic disorder. It is a right answer. It is a metabolic disorder and it is a lifestyle based disorder. Now, it is caused by the insulin resistance. That is also true. It occurs because when the body due to insulin resistance. So both and A are true, and the R is the correct explanation of the A. So A will be the right answer in this case. Now again we have to do match of the following. So enzyme means antibody plus enzymes. So then they are antibody having catalytic activity. Ribozymes means they are RNA molecule having catalytic activity. Coenzyme. So coenzyme is a part of an enzyme. So FAD act as a coenzyme for many enzymes, and lysozyme which is peptidoglycan of the bacterial cell wall. So lysozyme is present in tears or in egg white, in saliva, in sweat, all the secretions, and lysozyme it can cleave the bacterial cell wall, which is peptidoglycan. So according to this, right option A will be the right answer. Now the next question is, the association of endotoxin in gram-negative bacteria is due to the presence of and steroids, peptidoglycan, polypeptide, or lipopolysaccharide. So in gram-negative bacteria, LPS is present. So the right answer is lipopolysaccharides. Which of the following immunity is obtained during a lifetime? So there are two types of immunity: innate immunity and acquired immunity. Innate immunity is the one which is present since birth, and acquired immunity is the one which one acquired during their lifetime when one is exposed to some sort of when antigen or some pathogenic organism, and then their immune response, then their immune system mount a response against those pathogens. So that immunity is acquired immunity. Now next question is hepatitis vaccine. Hepatitis, it is a viral disease. Hepatitis vaccine is an example of subunit vaccine, killer vaccine, or the toxoid vaccine or the recombinant vaccine. So hepatitis vaccine, it is the first recombinant vaccine. I should write recombinant vaccine. So the first recombinant vaccine was developed against the hepatitis. So next question is which of the following is not a secondary lymphoid organ? So thymus, spleen, mesenteric lymph node, or Pierce patch. So thymus is not a secondary lymphoid organ. This is a primary. Bone marrow and the thymus, these are the primary lymphoid organ. Others, the spleen, lymph node, Pierce patch, all these are secondary lymphoid organs. Name of the class of immunoglobulin which has pentameric structure. I think this is very easy. No, IgM act as a pentameric structure. IgA, it acts exists as dimeric. All other exist as monomeric. Now next question, which of the following antibiotic contains a beta lactam ring in its structure? So the options are tetracycline, streptomycin, polymyxin, or penicillin. So penicillin, this class of antibiotic, they contains beta lactam ring in their structure. The next question is lectins are carbohydrate binding protein, lipid binding protein, DNA binding protein, or RNA binding protein. So lectins are those protein which binds to carbohydrates. So they are carbohydrate binding proteins. Option A will be the right answer. Lectins are carbohydrate binding proteins. So here I